You've probably heard of blood typing, and you've probably heard that when an individual gets a blood transfusion, we need to know exactly what bloods are compatible in that individual. So we're going to discuss blood typing. We're going to start from the beginning and go very slowly through blood typing so you can thoroughly understand why some people need to get certain types of blood. Blood typing revolves primarily around the red blood cells, since they're the most abundant cell in your blood. The red blood cells have, on their surface, a whole host of different proteins. These proteins are often referred to as membrane glycoproteins. A glycoprotein is just a protein that has some sugar molecules attached to it. In our discussion, we're going to refer to these quite often as antigens. And that might be a little confusing because you may have heard that antigens are foreign entities to your body, like a virus or bacteria. But these are not foreign to you. So why are they called antigens? They're called antigens because they could act as foreign entities when placed in someone that is not compatible. So when you hear the word antigen in our discussion, these antigens are proteins found on certain individuals' red blood cells. They're often also known as agglutinogens. I'm not going to call them that, but if you see the word agglutinogen, these are the same as the antigens. When certain foreign entities, antigens, are placed in individuals, that individual's immune system will make antibodies to attack and remove the antigens. Antibodies can be made against these antigens. These antibodies are often known as agglutinins. Again, I don't call them agglutinins, I will be calling them antibodies. We must understand the difference between antigens and antibodies before we go any further in our discussion. Again, antigens are found on the surface of red blood cells. Antibodies are defense mechanisms. They are proteins produced to get rid of foreign things from the body. If we have improper blood transfusions, what happens is an individual's antibodies will attack the new transfused red blood cells, destroying the red blood cells. This is called hemolysis. Upon hemolysis, the red blood cells release hemoglobin. This hemoglobin floods the bloodstream and must be cleared and removed by kidneys. The problem is this is a lot of protein for the kidneys to handle. This often will overwhelm the kidneys and lead to kidney failure. So improper blood transfusions are extremely dangerous for the survival of the patient. We're going to be discussing blood groups that are based on the presence or absence of certain antigens. There's two very common blood groups. The ABO blood group, which is based on two antigens, A and B. So individuals might have A, they might have B, they might have both A and B, or they might have neither A nor B. The other blood group we're going to talk about is called the RH blood group, which revolves around one more antigen called the RH antigen, or sometimes it's called the D antigen. Looking at the chart, we'll see that some blood types are very common in certain populations. Other blood types are very rare. Notice the two most common blood types in most populations are O positive and A positive. A lot of the other ones are very rare in populations. like B negative and A negative. So most of you listening to this video are either O positive or A positive. Doesn't make you any better. It just has to do with the frequency of the genes that code for the A, B, and Rh antigens. Okay, let's look at this picture. This picture sets the stage for understanding blood types and compatibilities. You'll notice this table only divides blood into 4 types. It does not include the Rh blood group. But if you think about it, the Rh blood group, you either have Rh or you don't. If you have it, we call you positive. If you don't, we call you negative. So that would make these 4 blood groups double into 8 total blood groups, because you could be A positive or you could be A negative. You could be B positive or B negative. You could be AB positive or AB negative. You could be O positive or O negative. The first row we've already done. It's showing a red blood cell and what antigens might be on the surface. So if you're type A, you have the A antigen. If you're type B, you have B. If you're type AB, you have both A and B. If you're type O, you have neither A nor B. So O is not an antigen. O just is in reference to neither A nor B. Okay, that's fairly easy. The more important thing is to understand what type of antibody would these individuals have the ability to synthesize. If you have type A blood, the last thing your immune system would do is to produce an antibody to A, because it would attack its own red blood cells. But it doesn't have B. Therefore, B is foreign to people with type A blood. And since B is foreign, individuals with type A blood have the ability to make anti-B antibody. This antibody is used to attack and get rid of the B antigen. So the last thing we want to do is put blood type B into someone who has blood type A. Same is true for B. If you have type B blood, that is because you have the B antigen. If you have type B blood, your body can make anti-A. Type AB has both antigens. Therefore, their immune system will not make neither antibody. The question I ask you is why? The answer is easy. It would be very stupid. It would not be a smart thing for this person's immune system to produce either antibody A or antibody B, because it would attack its own red blood cells. Type O has neither antigen, so it produces both anti-A and anti-B, because A and B antigen would act foreign inside this person who has type O blood. So we really need to go through steps to understand blood types. First, we make sure we understand what antigen the person might have. Second, we work on understanding what antibody that person has. Third, try to understand why certain blood will work in someone else and certain bloods will not work. For instance, if we have an individual who needs blood and they are type A, we cannot put B or AB in that person because of that B antigen. We can put type O. We can give someone with type A, type O blood, and all is good. Can we do the opposite? No. Someone with type O blood, we cannot give type A. We can't give type B. We can't even give type AB. So individuals with type O blood can only receive type O blood. The next thing we're going to work on is understanding positive and negative. That has to do with the Rh blood group. The Rh blood group revolves around an antigen, often designated letter D or simply Rh. If you have this antigen, you are positive. If you don't, you are negative. Just so happens most populations, for example Americans, are type Rh positive. But there is a subset that are negative. Rules still apply that if you have the Rh antigen, you do not make the antibody. And if you do not have the antigen, you will make the antibody. The difference is, for Rh, you don't make the antibody unless you're exposed to the Rh antigen. We call this sensitization. Whereas with the AB, it's spontaneously produced early in your life. So you have these antibodies, even though you've never been exposed to the A or B antigen. Production of anti-Rh antibodies by an Rh negative individual will cause hemolysis, just like the incompatibility that might occur with the ABO system. The reason why we study Rh is we found that there could be a problem when we have a pregnant mother who is Rh negative carrying an Rh positive baby. These bloods are incompatible. But during the first pregnancy, bloods don't mix. At the placenta, blood does not mix between mother and fetus. Nutrients get exchanged, waste gets exchanged, but blood does not mix. But what happens at birth is mixing of blood, which exposes the mother's immune system to the Rh antigen. We found that during a second pregnancy, the mother's immune system can pass antibodies into the fetus and destroy the fetal blood cells. This is called hemolytic disease of the newborn. It occurs if blood from an Rh positive fetus makes contact and gets inside of the Rh negative mother. This happens during the process of childbirth. Blood mixes when a baby is born. The mother's immune system would synthesize the anti-Rh antibodies and be sensitized. Upon being sensitized, a second pregnancy is the problem. During a second pregnancy, these antibodies can cross through the placenta and attack the baby's red blood cells. So in a review, let's go back here. This is a pregnant woman during her first pregnancy. She is Rh negative. We find that out by doing a routine blood test. The baby is Rh positive. We don't find that out. We do not test the fetal blood. We just assume that if she's carrying a baby, there's a good chance it's positive, because the father, if he is positive, the baby will be positive. Now, during this first pregnancy, there is no concern. The concern occurs at birth. So let's pretend this woman is delivering that baby. Blood vessels break within the placenta, and the Rh positive blood gets into the mother's. The mother produces antibodies. The baby has been born, so this first baby we are not worried about. But the mother is now producing antibodies. If she was to get pregnant again, those antibodies are small enough to cross into the fetus and attack those fetal red blood cells. In order to prevent this, there is a drug called Rogam. Rogam is actually an antibody. It's an anti-Rh antibody, and it's used to mask these antigens that have leaked into the mother during that first pregnancy. Rogam is given usually somewhere around the 26th to 28th day of pregnancy, that first pregnancy, and then shortly after birth of that baby. Rogam will block all these antigens, She does not make the antibodies. And the second pregnancy, that baby is protected. But all pregnancies, this woman will have to get injections of RhoGAM. But remember, it's only a concern when the mother is Rh negative and the baby is Rh positive. We can determine blood type very easily. We mix blood with the antibody. So for example, if someone is blood type A and we mix that blood with anti-B, it will not clump. If we mix it with anti-A antibody, it clumps. So wherever we have this clumping, called agglutination, we know that there's the presence of the antigen and the antibody. Someone with blood type B will agglutinate in the presence of anti-B antibody. Someone with AB will agglutinate in the presence of both anti-A and anti-B. And then finally, someone with O, their blood will not agglutinate with anti-A or anti-B antibodies.